So we are out in downtown Miami, right next to Fisher Island and right off the shores of Brickell, looking for sharks. So this is a really special project because we are fishing at night. Normally our research occurs during the day, so this is really exciting and new for us. My master's research, investigates the effects of artificial light at night on sharks. So I'm taking the blood from each of the sharks we catch and extracting the hormone melatonin. Just like humans and many other species of animals, sharks also have melatonin. Melatonin helps to regulate many biological processes like circadian rhythm, reproductive cycles, and growth, just to name a few. Using melatonin, I can compare the levels between areas of high light, like the one we're in tonight, to areas of lower light, like areas outside of the city, to see if this artificial light at night is really affecting sharks. Very little research has been published on artificial light's effects on sharks, and none, to my knowledge, have investigated the effects of ALAN on artificial light at night on melatonin levels. So this is really exciting. So to catch the sharks, we will be using drum lines. So drum lines have a 45-pound weight that just sits on the seafloor. Attached to this weight is about 100 feet of monofishing line terminating with a circle hook. So we like to use circle hooks because they are designed to hook the shark just in the corner of the jaw. This creates a very superficial wound that will heal very quickly. The circle hooks have a smaller chance than J hooks of foul hooking the shark. Also attached to this 45-pound weight is 30 feet of braided line that ends in 2 floats so we can find those drum lines again. The lines sit in the water for about an hour, and then we pull them up, check to see if anyone's home, and if not, we rebait and deploy them again. If we have a shark on, we then go into our workup. So on these types of trips, we typically set around 50 lines, so lots of weight. If we are lucky enough to catch a shark, the workup involves 3 standard length measurements. These length measurements will allow us to compare sharks and shark populations with other scientists. We also take a set of morphological measurements to see how girthy or fat the sharks in this area are. We can then use a combination of this length and morphological measurements as a non-invasive proxy for the health of the sharks. We also tag the sharks with identifier tags and take numerous biological samples from the animals. Once we have collected all of the data, we remove the hook from the shark's jaw and then watch it swim off. So we have many different types of tags at our disposal, but the ones we're using tonight are just identifier tags. So one of these comes from NOAA, or the National Oceanic and Atmospheric Administration, as part of their cooperative shark tagging program. So if a shark is recaptured by us, another scientist, or a citizen scientist, we can relay that tag number to NOAA, and then we get an email telling us when, where, and how big the shark was during the second capture. The other type of tag we use tonight are called pit tags. So these are subdermal tags that are also identifier tags. They last longer than the NOAA tags, so we can see if we catch the same shark multiple times. So some of the biological samples that we take are fin clips, a small muscle biopsy, and whole blood. We use the fin clips right now to look at DNA. So we're working with a collaborator at the Field Museum in Chicago, and he's looking at how genetically connected shark populations are in the US. In the past, we have also used the fin clips to investigate toxins such as BMAA and methylmercury that can be found in sharks. We can also use these fin clips to determine the trophic level of sharks' diet, so we can see kind of where sharks are fishing in the big food web. We also take a small muscle tissue and send it to a collaborator in Brazil who is looking at fatty acids in sharks. So these fatty acids help researchers determine the shark's energy stores, which can then tell us about the health of the sharks in the region. And finally, we take blood samples. We collect whole blood from some species. From this whole blood, we can also determine the trophic levels of their diets, but on a much shorter term scale than the fin clips. We can also use the whole blood to determine how stressed sharks are using lactate and glucose meters, just like we can in humans. We can also, from this whole blood, look at their hematocrit, or their percentage of red blood cells in the blood. So this hematocrit will tell us the oxygen carrying capacity in the shark's blood. We can then spin this whole blood down into plasma and look at nutritional hormones such as triglycerides and fatty acids. This will tell us, again, some of the health of the sharks in the area. This plasma can also tell us about reproduction. We send plasma to researchers in Arizona who can then tell us if sharks are mature. So we can compare that to the length and maturity, so their total length that we measure. Or we can also see if the sharks are pregnant. How cool is that? We can tell if a shark is pregnant. Some of the plasma we collect tonight will also be included in my project. And so again, my project, I will be analyzing it for melatonin, the melatonin hormone. We caught 5 sharks tonight. Woohoo! We got 3 black tips, 1 nurse, and 1 lemon shark. What a wide range of species. That's so incredible. We were actually able to collect full workups on all 5 sharks. So this is great data for not only my project, but also all of those other projects that we send those samples to. So for my master's research, I'm just trying to get a baseline to see if sharks even really care about artificial light at night. It really is just a pilot study. And then down the line, if we do find these harmful effects of artificial light at night, we can think about different light regulations. So something like they have for sea turtles. So instead of white light, we do red light. But we are very, very far from any of those big policy changes. Again, this is just a very basic pilot study.